In this video, we will discuss transcription in prokaryotes. Here are the 4 steps that we will discuss. RNA polymerase is going to be our main enzyme in this process. When the RNA polymerase core enzyme associates with the sigma factor, it is called a holoenzyme. Its primary job will be to recruit the RNA polymerase to the start site of transcription. Let's let this represent our DNA strand. This is the +1 nucleotide where transcription will start. Upstream from that, we have our promoter region. There is a -10 TADA or Pribno box and a -35 control region. This is the rest of the DNA strand. Here is our RNA polymerase with the ends of the wires representing the sigma factor. As the holoenzyme comes across the promoter region, the sigma factor recognizes and binds to the 2 consensus sequences on the DNA. After the transcription of about 9 to 12 nucleotides, the sigma factor actually dissociates from the RNA polymerase, and transcription can now continue. Here we have a question about a 5 base pair insertion between our 2 consensus sequences and its impact on a gene that codes for an enzyme required to produce the amino acid phenylalanine. Let's add 5 base pairs between the -10 and the -35 sequences. Now let's bring in the sigma factor. So sigma factor cannot recognize the promoter sequence, RNA polymerase will not bind to the DNA, transcription will not occur for the gene, and phenylalanine will not be produced. Phenylalanine is used by the body for sending signals throughout the body, and a failure to produce it will cause further complications. Once RNA polymerase binds to the DNA strand, the DNA unwinds, creating a transcription bubble. This is where transcription is occurring. DNA is unwinding in front of it and rewinding behind it. Here we have our black and green DNA strands and the gray RNA strand that is being transcribed. RNA transcription occurs from the 5' to the 3' end. This means that DNA is being read 3' to 5'. The bottom strand is known as the template strand. The upper strand is the non-template or coding strand. It goes in the same direction and has the same base pair order as the RNA strand, except thymine is replaced with uracil. These bases are added to the growing RNA strand, and by the cleavage of 2 phosphate groups from RNTPs, RNA polymerase forms phosphodiester bonds 5' to 3' to connect these nucleotides together. Given this sequence of bases on the template strand of DNA, let's fill in the complementary RNA strand bases. Inverted repeats like this one are present on the DNA sequence near the termination site. When transcription occurs, a hairpin will form on the RNA strand. This will cause the RNA polymerase to pause during transcription. In row-dependent termination, the protein row attaches to the growing RNA strand and its helicase activity causes the DNA-RNA complex to separate. Think of it like scissors. In row-independent termination, the hairpin is followed by a tail of U's. The AU bonds are weak and allow the dissociation of the DNA and the RNA. That brings us to the end of prokaryotic transcription.